कभी-कभी ट्रैवलिंग में होता है।
सर ऐसा नहीं है कि सर आपको सुनना नहीं चाहते सर, ऐसा नहीं है, अभी थोड़ा और 5 मिनट बाद ज्वाइन कर लेंगे सर वो लोग, सबका ऐसा ही होता है। हाँ
ये सब बहाने ये ट्रैवलिंग स्टफ ये मैं नहीं मानता।
आजकल नया जमाना है, ये बुड्ढों का भाषण नहीं सुनना चाहते। अब हमको डॉक्टर डॉक्टर मेहरा को सबको रिटायर कर दो।
Sushant, also please check out. There is echo.
Yes, ma'am. We can start.
नहीं echo हो रहा है voice. जरा देखो कैसे हो रहा है.
Echo.
I think there is some echo. Maybe
जी, sir.
somebody has joined from more than one source.
Yeah.
Yes, sir. Yes.
Generally, if somebody in the group has joined from more than one source.
May I request everyone to be muted so that we will stop hearing the echo?
Yeah, now it is okay.
Yeah. Thank you, sir. I welcome all the Indian Immunology Society members to the Pratiraksha Swarna Jayanti Vyakhyan 2 by esteemed speaker, Professor Dr. N.K. Ganguly, Padma Bhushan by the President of India. Ji sir, Nirmal Kumar Ganguly. Padma Bhushan by the President of India, former Director General, Indian Council of Medical Research, Chairperson, Immunology Research Foundation, and more details on his achievements would be highlighted by Professor Mehra, who is chairing today's session, INSA Vice President of International Affairs, and I will briefly introduce Professor Mehra. Sushant, kindly display Professor Mehra's introduction slide. We thank Professor Ganguly for joining us. He's shortly back in India from his US trip, and still he obliged us by accepting our invitation to give this second lecture of the Indian Immunology Society Golden Jubilee lecture series. I am really honored to read this introduction for Professor Mehra, Professor Narinder Kumar Mehra, Vice President, Indian National Science Academy International Affairs, recipient of prestigious Shanti Swarup Bhatnagar Prize for Science and Technology, and the Chevalier of the National Order of Merit from Francois Mitterrand. Former Dean of Research AIMS and ICMR, Dr. C.G. Pandit National Chair, elected Fellow of the International Medical Sciences Academy, the World Academy of Sciences, Indian National Science Academy, and National Academy of Sciences, India. Professor Mehra is renowned for his research on histocompatibility and immunogenetics. Founding Secretary General of the Federation of Immunological Societies of Asia Oceania, which we know as PHIMSA, Council member of International Union of Immunological Societies for more than 12 years, and organizer of the several advanced courses on basic and translational immunology conducted by PHIMSA in collaboration with IUIS and Indian Immunology Society. He is president of Indian Society of Histocompatibility and Immunogenetics, member of the National Board of Advisors for the Center for Stem Cell Science, the Advisory Board of Indus Foundation, Publication Advisory Board of INSA, and a trustee of the Board of Immunology Foundation. A recipient of several national and international awards, H.A. Mehta Gold Medal, ICMR Shakuntala Amirchand Prize, Sher-e-Kashmir Sheikh Mohammed Abdullah Award, Ranbaxy Science Foundation Award, ICMR Jalma Trust Foundation Award, Om Prakash Bhasin Award, Chief of the Army Staff Award, ICMR Basanti Devi Amirchand Prize, Khwarizmi International Award, and Dr. B.R. Ambedkar Centenary Award. These are some salient ones that I wrote, sir. Over to you, Professor Mehra. Please take the proceedings. Thank you, sir.
Thank you, Taruna, for those very, very kind words. It's always nice to hear about you, about yourself so much. And of course, we have had a long journey in science. I started doing science in the early 70s. And that is when, you said a lot about Dr. Ganguly. one thing is also needed to be said is that he's the founder of the Department of Experimental Medicine and Biotechnology in Chandigarh. He, of course, moved from one department to the other, and then he's the founder of that. And that department
So
is
may I interrupt you for a minute? Sushant, yeah, he has displayed sir's slide. Yes, thank you, sir. Please proceed, sir.
That Department of Experimental Medicine and Biotechnology in EJ Chandigarh has done great service to most of the researchers over there. And I would say that it has really encouraged a lot of people from the clinical sciences to do research. My own association with him goes back to about 1976, when Dr. Wahi, who was the then director of the EJ Chandigarh, a very renowned cardiologist, and Dr. M.L. Bhatia, the cardiology head in AIIMS, decided to do a one-on-one meeting in Chandigarh to discuss about rheumatic fever and rheumatic heart disease. And we were a group of four or five from AIIMS, including Dr. Malviya, Srinath Reddy and all. And I do remember that time Malviya told me, यार थोड़ा सा पढ़ के जाते हैं अच्छी तरह से, we have to make an impression about AIIMS. तो हम खूब पढ़ के गए थे, और उधर चंडीगढ़ साइड से इनको हम जानते नहीं थे, और ये जो भी हम यहाँ से वॉली फेंकते थे ये उसका जवाब देते थे, बड़ा जबरदस्त जवाब देते थे. तो उस दिन से हमारी दोस्ती शुरू हो गई, and by God's grace we are very good friends since that time. So you can say it already goes to more than 45 years. But one quality I must tell to the younger audience over here, and I think they must learn from his style of living. Is there somebody? Is there anything? Are you able to hear me?
Yes, sir. Yes, sir.
Okay. So I was just trying to highlight one quality of his, and that is he wakes up in the morning at 4 a.m. every day. And this has been doing for the last maybe 50 years. And he reads a lot. You know, a lot of people get surprised, how does he develop so much of knowledge? And that is his reading habit. He reads at least 2 to 3 hours a day, not only regarding his own field of research, but all about medicine and biology. And I think this is a very important habit that every youngster should develop. Reading, gathering knowledge, and gathering knowledge just not in your own field of research, but broad, because you never know when it will come. I do remember sometimes, there was a conference going to be held on psychiatry, and the secretary head in AIIMS asked me, whom should we call as the chief guest? I said, call Dr. Ganguly. He said, but our conference is on psychiatry. I said, you will be surprised. He will know everything about psychiatry. Because that's the quality he has. He gathers knowledge and he works hard. Even the knowledge doesn't come on its own. You have to work hard for it. And so he keeps himself updated, and sometimes we all get surprised when he comes out with just a 1 week old concept. And we can see that this concept comes from his habit of reading, and he continues with this habit even till his date. And you can see this is from his today's lecture talk. It's all about immunophenotyping, immunotherapy, checkpoint inhibitor therapy. This is not the field that he has been working all his life. Maybe in recent years he has been multi-omics and all that. And he's 80-year young and still going very strong. And you can see that he keeps himself updated with all that is going on around the world in the field around immunology and biotechnology and the technological field. He has, his paper numbers, you don't have to talk about them, must be about 800 or so. His awards go into more than 100 or 150 there. He has been a fellow of almost all the academies, and becoming a fellow of the academy and of all academies itself is an indicator how remarkable a scientist he has been all his life, because these fellowships don't come from nothing. Actually, you have to really contribute significantly for others to note and therefore recommend you for that. He has been chairperson of many institutions in India. He has guided many institutions in India. He has made careers of several young scientists. This all goes without saying. And yet he remains very humble and grounded. The other good quality that he has is of forget and forgive. I think this is a very good quality that everybody should develop. As you age, you have to develop this habit of, because there are a lot of people who will really come around you, and you may not like this and this. The best is to forget and forgive and move forwards. So these qualities I would like the youngsters to imbibe from his own career, besides the good quality science that he's done all his life in various fields of immunology. And today you're going to hear about his latest love for immunotherapy. So I really don't want to stand between the audience and the speaker, and I give the floor to Dr. Nirmal Kumar Ganguly. You can see that we both share initials, and there was one time a symposium on NK cells, and we were both speakers over there. People were calling us also as natural killers. Thank you so much, Dr. Ganguly. Here is the floor for you. Please go ahead and give your lecture.
Thank you very much, Mehra Bhai. And we have been friends for now more than 4 decades. And it is, I'm very delighted that he's sharing this particular session. I start for the Immunology Society members with one of the Kabir's Doha. मैं बुरा जो देखन जग चला बुरा न मिलिया कोई जो दुख जो मुंह देखो आपनो मुझसे बुरा न कोई. We need to remember this in all our activities and function in the future. Move to slide. Yeah. So these are the contributors to this particular talk. The first one is Kriti Jain. She is my PhD student. Then Amit Awasthi, Dr. Amit Awasthi is an immunologist. I use his equipment and he they also gave me a lot of reagents. Dr. Sham Agarwal is the clinical oncologist, a medical oncologist in the Gangaram. Then Deepak Rathore works with the Amit Awasthi. He helped in the analysis. Bhavtosh Das is the PhD professor. He helped in the microbiome. Jaswinder Singh is just popularly known as Jassi, was in the THSTI, and he helped in the metabolomic. And then we have Professor Surjit Ganguly from the Jamia Hamdard. He helped in the inflammatory pathways, and Parul Chug helped me in the statistical analysis. So my main question, what I asked was, to the immunology audience, I need not say what is checkpoint inhibitors or what is PD-1 or PD-L1 and how do they do. Essentially, the immune cell should recognize an antigen, or in this case, it is tumor. However, they develop, and there are two signals. One is the CD3 signal, primary signal, and there need to be co-stimulatory signal which actually complete the immunologic stimulation, and ultimately responses come. So here they create ligands which look like co-stimulatory molecules, so to evade the recognition. And that's why the immunologic cells which do not get primed to tumor cells, and they cannot attack there. So PD1 is a T cell inhibitory molecules, and CD8 cytotoxic T cells need to recognize the recognize the cells and this actually prevents it. PD-L1 is the PD-1 binding ligand of tumor cells, and here the APCs and macrophages need to recognize, need to interact with this. So the PD-1 PD-L1 interaction normally results in T cell suppression and energy exhaustion and death occurs. So blocking the PD-1 or PD-L1 pathway will promote the function of exhausted tumor-specific T cells and decrease tumor-induced immune suppression. So this is the pathway which is important. So So our main aim was that only drawback in this the checkpoint inhibitor therapy normally helps into this, and it helps into this ligand binding, and thereby the immunologic cells could interact with tumor antigens and thereby respond. But drawback is only 30 to 35% are responding to this therapy. So challenge is to develop a set of biomarkers to predict and monitor responses, as well as those who will respond to this therapy. And because the therapies are very, very expensive and people get pauper, they have to sell their home and hearth if they are given this and they are not likely to respond. At least those who are not likely to respond, you can use additional adjuvant or use other therapies. So the present biomarkers which are used is PDL1, the tumor mutation burden, and microsatellite instability markers. So, but many of these are non-specific with limitations, and that is why we cannot predict with certainty that this person will respond and this person will not respond. So identification of specific biomarkers to predict response is our ultimate goal for this study. To stratify cancer patient receiving PD-1 targeted therapy, so to stratify cancer patient receiving PD-1 targeted immunotherapy broadly in two categories, we have decided, we have done responders and non-responders. So those who are non-responders have progressive disease. Those who are responders may be completely responding, partially responding, or may go into a stable disease format. So our study group comprised of normal healthy volunteers. There are 13 number. The lung cancer 66%, and melanoma 10% subjects, oral cavity subjects 18%, and hepatocellular carcinoma 6%. Cancer patient receiving anti-PD-1 therapy was total number 50, and there were 3-point sampling of these. Now we had chosen those groups because the US FDA has declared about 6 cancers where the immunocheckpoint therapies could be given as a primary therapy. You need not give some additional therapy over there, because if you give additional therapies, it is very difficult to really dissect the immune responses. So we only chose those cancers where the immune checkpoint inhibitors could be given primarily. The majority of our patients got nivolumab because it was the cheapest and it has got an additional therapeutic pathway compared to Pembro. Pembro also is a PD-L1 checkpoint inhibitor, but the Pembro is a little bit more expensive and it has a one pathway lesser in its interaction. So we in this study group, we essentially looked at some of the markers, immunologic markers. and the CD points are given over here. So we looked at monocytes, macrophages, dendritic cells, whether plasmacytoid or myelocytoid, NK cells, natural killer T cells, T cell subsets, TH, T cytotoxic, Tregs, T cell proliferation, naive, extended memory, central memory, TEMRA, T cell activation, stimulation, and cytotoxic potentials, and B cells. And the markers which we use are given on the extreme left side. So, so as I told you, they are given to only eligible persons, and nivolumab we gave at 3 milligram per kg every 2 weeks by infusion. Blood samples were collected before initiation and after 4 to 5 weeks, followed by at the T2 point and up to 14 weeks. RECIST criteria was used to document progression of the disease clinically. Response assessment was done clinically after each cycle and radiologically after 2 months of the therapy, and after every 2 to 3 months it was followed up. So we have about 5 years follow-up of this. Complete response, I will not read out everything over here, but disappearance of the target lesions and other pathological lymph node clear lymph node, etc. Partial response was at least 30%. Progressive disease was at least 20%, and stable disease was neither sufficient shrinkage or other markers were seen. Next, please. So the multi-omic approach was used to identify biomarkers. So the first material was PBMCs. It was used for immune profiling, and we used the BD FACS area 3 as well as another equipment which was a little more advanced in THSTI. From the plasma, we looked at proteomes and we looked at metabolomes, and we used Orbitrap mass spectrophotometry. From the serum, we looked at the cytokines, and we essentially used Luminex, but we also used flow cytometry. For the tissue blocks, we used it for genomics. We used essentially targeted genomics at first, so we did. I have used iron torrent, but later on MySec was also used, and now some of the samples are being looked at through NovaSeq. For the fecal samples, we looked at the microbiome, and we used the MySec instrument. The comprehensive assessment of immune composition to predict response we did by flow cytometry. In T cells, we looked at helper T cells, cytotoxic T cells, NK T cells, gamma delta T cells, B cells, naive, memory, plasmablasts, and plasma cells, NK cells, cytokine producing cells, cytolytic cells, myeloid cells, monocytes, macrophages, dendritic cells for both classical, intermediate, non-classical, and conventional plasma cytoid cells. Then we looked at granulocytes, neutrophils, basophils, eosinophils, and mast cells. And in the T cell area, as I had said, naive, effector memory, central memory, regulatory cells. So there's how the samples were done. This was done, one was treatment sample, and after 2 to 3 weeks, when the PD-1 therapy began, post treatment at the T1 stage, and after 2 to 3 weeks, post-treatment T2 stage, and ultimately up to were collected at 3 time points as per treatment cycles. It was very, very difficult, but we did that. Next, please. These were the gating principles of the samples. I did not elaborate on this for the enlightened community, but this is how we used about 5 panels. The panel 1 was for the T cell and its subsets, And as you see that these were the 8, we used 8 markers and we used 8 monochromes. So 8 colors flow cytometry were done. Next. So if we really look at this, so if we look at this, then we can see that CD4 T cells were enhanced post-therapy in responders. Next, please. If we looked at the CD4 plus Ki67 plus T cells which were responding T cell which actually caused the damage, then CD4 plus Ki67 plus T cells enhanced post therapy in responders. The upper panels show the details, so I'm avoiding the details for the time's sake. Then for the CD8 T cells, they also enhance post therapy in responders. And when we looked at the CD8 when we looked at the CD8 plus Ki67 plus PD1 plus T cells, the post therapy it enhanced in responders. So CD8 extended memory cells, central memory cells, we didn't find much significant correlation. Next, please. These are the details. And then panel 2 was NK, NKT, INKT, gamma delta T and Treg cells. And these were the again the 8 CD panels and different fluorochromes. So NK cells increase post therapy in responders. So NKT cells, next were NKT cells. And gamma delta cells increase post therapy in non-responders. So this was increasing in non-responders. So the panel 3 was dendritic cells and monocytes. We found some important thing over here. The dendritic cells increase in the post-responder, but it was the plasmacytoid dendritic cell, not the myelocytoid dendritic cells. Next, please. The inflammatory monocytes increase post-therapy in non-responders. That is understandable because these are the ones which suppress the response. Now the CD8 Ki67 plus cells, they increase post-therapy in responders. This was panel 4. And in the same way, the CD8 plus ganzyme B cells increase post-therapy in responders. That shows that effector cells here. We did, we have another set of data which I'm not showing here. That was the genomic, transcriptomic, proteomic combined set, which is done by a new platform which is known as Olink. So we did it in Karolinska Institute, and it confirmed that the ganzyme, there was ganzyme, as you know, there are ganzyme 1 and ganzyme 2. With the immunologic responses, we cannot look at ganzyme 1 and ganzyme 2 separately. It comes as a total ganzyme. So we could dissect it and found that ganzyme 1 is the one which is affected. So this was the final one. Beta, humko bolne diya. So these were the final 5 B cell subsets. Next. In the B cell subsets, आगे चलो, ये तो details है. In the B cell subset, we didn't find any significant changes. So if we look at the summary, plasmacytoid dendritic cell enhance antigen presentation to T cells, and they increase post-therapy significantly in responders. Myeloid dendritic cells, they cause T cell suppression, promotes tumor growth, and post-thello I'm listening to some sounds.
I'm so sorry, sir. Sushant, please mute Dr. Vignesh. Sushant, please mute Dr. Vignesh.
So myelodendritic cell which supplies tumor growth cells were decreased. NK cell release cytotoxins directly in tumor cell levels were significantly increased post-therapy. So in the responders, new antigen specific lymphocytes, that CD8 plus PD1 target tumor specific new antigens significantly increased ICT in our responders. So this shows that actually the PDL, the therapy worked because they were able to recognize them. Blockade of PD1 pathway induces proliferation of effector-like cells. So this also happened. PD1, CD8, Ki67 T cells increase significantly in responders. And we need to really set up a limit to this, otherwise it will be difficult to really look at that what's happening. So our limit was because it will be difficult to interpret in a single patient. About fourfold increase in Ki67 CD8 T cells happened in responders. So granzyme is the indicator for increased cytotoxic potential of T cells. This also happened. This increased post ICA. Inflammatory monocytes possibly indicative of tumor metastasis that increased in non-responders post ICT. So now we did the cytokine profiling. I will not go into details of how we did it, but these were pre-mixed multi-analyte kit, and this was done in the Luminex platform, and we use the THSTI Luminex platform over here. We ultimately, we wanted to do more, but we ultimately did 14 here. But in our O-Link platform, where you could do more with the gene responder thing, we ultimately did 48. So, but here I will be present. That data is still getting analyzed, so I will be present to you the results of these 15 cytokines. So this is the heat map, and as you see, this is the baseline sample. The red is non-responders and green are responders. You see most are the reds at this given time. Next, please. And this is 8-week post. The reds are turning into in those areas where they respond, they're turning into green. Next. Then there's a 12 weeks post ICT. Next, please. So these are the result, complete analysis cytokine profiling. Some surprises came up. IL-23 increased at baseline in responders. And the secretory PDL1 and secretory and IL-21 increased in responders post. So these are very significant findings. And these were not really reported except like the recent report of IL-23 after the checkpoint inhibitor therapy. Next, please. So we propose a composite biomarker using secretory PD1 and secretory PDL1 to predict nivolumab efficacy in NSCLC patient with a longer OS. So this was the overall activity in the nivolumab samples. We have up to 12-week only, but we are now planning a larger study in the follow-up samples. So this shows the genomics, NGS, the tumor mutation burden, and microsatellite instability analysis. I will skip the how we process the samples, etc. So, but I will show the results of the targeted genomics also only at this given moment for the time. So the TP53 gene mutation can be associated with ICI benefits. 62.5% responders, partial or complete, had this genomic alteration. FGF4 gene mutation, 48% of responders showed this genomic alteration. TP53 is a tumor suppressor gene. The TP53 gene is the most frequently mutated gene in human cancer, indicating that the TP53 gene plays a crucial role in preventing cancer formation. P53 gene encodes proteins that bind to DNA and regulate gene expression to prevent mutations of the genome. So this was something very, very significant which we got. FGF4 is significantly associated with nodal involvement and overall survival in the patients with different types of cancer. So the tumor mutation burden we didn't find. Panel targets were 1.65 million total bases, out of which 1.2 bases are from the exonic locations. We looked at the 43 driver genes and mutations in 61 driver genes and hotspot mutation in 61 driver genes. We didn't find any significance in this. This is actually have been reported by people. So tumor mutation burden difference was a surprise. The results of tumor mutation burden is seen there. There was no difference in this. So this was a surprise that we didn't get it. Microsatellite instability analysis, again, we didn't get. I will skip the details. We didn't get any significance in this. So after this, I go, so three established one we didn't get. We found two targeted gene mutations but this was after filtration of thousands and thousands of genes. And so, because to conserve time, I have showed you the positive results only. The immunohistochemistry we did with the PDL1. Again, PDL1 testing on tissue selective half-wave rockets were done. The conventional methods as well as the conventional monoclonal antibodies were used. You could see the difference in the HEZ and PDL1 thing. We found the PDL1 expression varies in cancer types, but more than 1% PDL positive was interpreted as positive. If you get 10% or more or 30% or more, the responses change. So this is how the PDL1 expression happened. PDL1 expression positively correlated with disease outcome in responders versus non-responders. It was significantly high in responder subsets. We found one more marker, and out of the many markers we looked at, this was desmocollin 3. It has a tumor suppressive activity through inhibition of AKT pathway. AKT pathway, as you know, is one of the important pathways in tumors, and some drugs on AKT pathway are available and being used in cancer. So here we see that more than 50% desmocollin 3 expression is taken as a positive one, and we found in the responders the desmocollin 3 expression increased. Now the plasma metabolomic study, this is the study design. I will skip this and we'll go to the two major analytical method: principal component analysis and partial and the partial component least square determinant analysis, which is known as PLSDA. So if we look at the principal component analysis, we see the green one is the healthy controls and the pink one is the cancer. We can see that, and we took ultimately the baseline in the lung cancer patients with healthy control because they were the highest number. So from now onward, I will be presenting mostly data on the lung cancer. So this is the volcano plot, and you can see how the controls and the cases are separating beautifully. There's the heat map of hierarchical clustering of 50 top metabolites. I will identify the metabolites in the next KEGG analysis. And you look at the top one, the glutathione and glutamate, alanine, aspartate, leucine, nicotinamine, taurine, etc. These were, so there were many of these. We took 50 metabolites, and enrichment overview is given for top 25 metabolites. Next, please. So this was the KEGG and path analysis down-regulated metabolites, and down-regulated metabolites were caffeine, the thyroid hormone, the catecholamines, etc. And this is volcano plot analysis as metabolite non-responders versus responders. And you could see a clear-cut separations in the responders and non-responders in this KEGG analysis. This is again the principal component and partial least square determinant analysis of the two group for cancer based on metabolic profile of patients. And you could see how these two are nicely being clustered and separated. So this is a heat map of hierarchical clustering of top 50 metabolites, which are capable to segregate non-responders, red bar, from the responders, which is green bar, and baseline the black ones are actually not changing. So this is the KEGG pathway for the upregulated metabolites. I will come to the next slide and describe the major pathways. So we found three major pathways ultimately, which are significant. Sphingolipid metabolism, arachidonic acid metabolism. The sphingolipid induces tumor cell proliferation, resistance to chemotherapy, radiotherapy or immunotherapy, and metastasis. Arachidonic acid path metabolism, is one of the major metabolic pathway for inflammation. And glutamate metabolism, actually this reconditions the tumor microenvironment and can enhance the anti-cancer immune response through both direct and indirect mechanisms. This particular kind of thing has been published by Young in the General Cell. So KEGG pathway analysis of down-regulated metabolites. I skip this, but go to some of them. As the Kriti actually skipped few of the slides, but I, in respect to her, I come to the major metabolites. One was the urea pathway. You can see the difference. Sphingosine pathway, you again can see the difference. 4-hydroxybutadiene pathway, again very significant differences are there. And Gentisec pathway is again showing clear-cut differences. So this is the glutathione pathway, and there is a general cancer treatment, metabolism, progression and treatment resistance has been published in JCB. Urea cell cycle dysregulation, again, clinically relevant genomic and biochemical signatures have been published in the recent Cell paper. So these are the ROC curve for the urea. Next. This is the combined ROC. We have about 100% sensitivity is obtained in this, and specificity is around 67%. Is the Kaplan-Meier analysis, 5-year progressive survival in lung cancer patient. You can see the difference very nicely in the Kaplan-Meier. And this is the progression-free survival in lung cancer patients by plasma level. And this is the 5-year-old survival in lung cancer plasma level of urea discovery and validation set. We actually again validated it. The discovery part we took to validation so that we know that it really works. So this is the survival curve, and you see that this really survival curve is very significant. Clinical correlation plot with the associated biomarker panels, and I will show the results in the next one. You could see lung cancer, and that the clinical survival plot will go back. The clinical survival plot for the various metabolites was associated with the biomarker panel. So this was correlating with the biomarker panel. Next, please. So now we looked at the microbiome analysis. In the microbiome analysis, in the responders and non-responders, we found the alpha diversity analysis actually, which looks at the observed O2U, evenness, Shannon index, and Chow index. we found that there was no significant difference, and we applied Wilcoxon test in this. Next, please. So this is the p-heat map showing the differentially abundant taxa between responders and non-responders. We find that really there was a differentiation of these taxa. I will come to the real bugs later on. So this is the bugs. There's a box plot. So we see the bifidobacterium is abundant in responders and not there in the non-responders. We see Klebsiella, which is a nesty worm, is high in the non-responders and is not found in the responders. The Doria, which is high in the responders and not found in the non-responders. So the Enterobacter was found in the non-responders, and there is a new ST taxa which was found in the responder and not found in the non-responder, and porphyromonas were found in the responders and not found in the non-responders. So if you really see the microbiome, which actually are becoming very, very significant. There are data in colon cancer, there are data in other cancers, and we see that it alters significantly in the non-responding patient, giving some kind of a hope that if we change them through these stool transplantation, fecal transplantation, maybe some of those who are not responding will respond in the future. Next, please. So this is the differentially abundant core functional pathways for which have been correlated with this. Next, please. Next. Next. So this is the map where we could really show the bug as well as the metabolic pathways which are uh, tweaked by them, and we see that the bugs are not there simply as a side watchers. They are actually participating. Next, please. So whether it is urea cycle or and the same, you see there's a beautiful correlation between the responders and non-responders and the bug. So to summarize the whole thing, the predictive biomarkers for response and OS in the patients treated with immune checkpoint inhibitors. For the immune cells, decreased T lymphocytes, B cells, activated T cells, DCs, NK cells, Ganzyme B, T cell proliferation, the inflammatory monocytes, increased T lymphocytes, DCs, the plasmacytoid DCs, activated T cells, T cell proliferation, Ki67, monocytes, NK cells, Ganzyme B expression, baseline CD8 plus cells. In the cytokine expression, we got CD23, the PDL1 decrease, IL-223 increase, and in the responders, IL-2 increased, IL-23 at baseline increased, IL-18 and PDL1 increased. The protein expression, tumor PDL1 and desmocoline 1 decreased in the non-responders, and expression of PDL1 and desmocoline 3 increased in responders. Genomic signatures, no significant correlations within tumor mutation burden, and microsatellite instability was seen, and tumor mutation burden, TP53 and FR4 gene increase. Metabolic signatures, sphingosine, butanol, glutathione, gencitic acid, sphingolipid, and metabolic pathway. All of these actually increased in these non-responders. However, the responders had a very nice urea pathway, arachidonic acid pathway, and glutamate pathway. Gut microbiome, Enterobacter, Klebsiella, xenobiotic deregulation pathway was seen in the non-responders, and Bifidobacterium, Doria, Porphyromonas, Fem family, and lysine biosynthesis pathway, which was linked to this, increase in the responders. So, to conclude, principal blood analysis can provide valuable insights into dynamic changes in T cell responses during PD-1 targeted therapy of cancer patients, favorable immune periphery at the baseline in responders, and this multi-omic approach along with comprehensive immune phenotyping may serve as a potential biomarker panel to monitor predict response to checkpoint inhibitor therapy. May stratify cancer patient receiving PD-L1 targeted immunotherapy broadly into categories, responders and non-responders. Faster immune response is also beneficial for the clinical response. This is showing one patient. This was no surgery was performed. You can see a huge cancerous lesion, and after therapy, this has disappeared. So thank you very much. And we have now about 30% or more patients surviving after 5 years. We are now looking at some of the markers, new markers, and looking at the transcriptomic as well as as well as the gene and proteomic signatures together. Proteomic data, I didn't show you the detailed proteomic data. Someday in the future, it is still being analyzed. There is a work in progress. And we are also looking at now complete whole exosome sequencing with the Kalyani Institute to really identify more mutation, because we are not satisfied that we didn't get total mutation burden or microsatellite instability markers in our study. Some people had found these as a positive marker in their study. So we are repeating them through the NovaSeq complete NGS of our exosome sequencing. So when we will get it, we will find this important. So I will like to tell our younger generation while finishing that you will go on working in the laboratories, whatever is your age, and it is the way, it is your responsibility to find your way in the laboratories. Mostly, government will not employ you after you have crossed 70 or something. But if you really want to be relevant in science, you have to find a method of go on working. Also, every few years, you have to change yourself to be relevant for the science which is developing in the area. Immunology is a very uh, nice and blooming science. Thousands of flowers go on blooming. When I was taking regular immunology lectures, every year I had to change my lecture because things changed. Some of the established dogmas, they fell down, and some new algorithms came up. So the persons who have taken immunology should be very vigilant that they are relevant for the future and go on updating their knowledge, because thousands and thousands of knowledge is coming up. And whosoever is doing multi-omics, everyone is looking at immunomes in much more detail than I have seen. I was a little bit limited with the money because all of this cost a huge amount of money, and like GP Talwar dependent on lot of donations of the reagents from the friends. But this is so huge now that in the translation, it will play a very big role, and particularly the multi-omic approach will perhaps found some future solutions. Thank you very much, Taruna, for providing me this opportunity. I hope I measured up to the brilliant new generation immunologists. Thank you very much.
Thank you very much. We had to all sit at the edge of our seat to imbibe it all. And you can see the enthusiasm. Javed Agrawala writes his age to be 83. Javed, he's 1 year less. He's still 82. He will complete 83 next year. He's entered 83. I and he are apart 8 years or maybe 9 years or whatever. But you can see the enthusiasm. The enthusiasm is as if a 35-year-old young man is speaking. And this is something that the youngsters have to learn. I want you to ask questions not only on the science that he showed you today of immunotherapy, but on his long career, because the Indian Immunology Society has started this Prati Raksha Swaran Jayanti Vyakhyan. And Taruna Madan and his team, on their own wisdom, has decided to pick up, these seniors and very seniors for these kind of awards. So I think, and rightly so, but because you can learn so much from their own careers. So you could actually ask him anything about how he started, how he came to immunology, what has he done, and how he keeps changing. And that was his last advice. I gave you already 2 tips from his career. Wakes up at 4 a.m., reads a lot, reads a lot, reads a lot. I think that's the key. And the second I told you was about his ability, his quality of forget and forgive and move forwards. And the third, he told you that be relevant in immunology, keep changing with the times.
So one of the things which I will like to tell to the audience, that never get intimidated with the bad luck which will dog you all the time, and get a teacher. If you get good teacher. So I was very fortunate that my teacher, Dr. S.C. Agarwal, was somebody who was a visionary. So Dr. S.C. Agarwal he did his MD from Lucknow Medical College, KG Medical College, and did his PhD in Oxford, and discovered for the first time that, but later on he was awarded DSc from the same university, found out for the first time the mechanism of action of diphtheria toxin, that how it interrupts the protein synthesis. So this is how he got me started. I didn't want to. I went to PGI to get into Department of Medicine, but I was having dinner with him, and he brainwashed me. He said all these Nobel laureates have come to this subject, so you take this subject, induced me to write an application, and gave me admission the next day. Those days, you didn't find many students in my subject. So when I went to join, so all the teachers there told me, you run away. There were 6 people before you, and they have run away within 3 days to 15 days. So don't waste your time. Run away from here. So it
Yeah.
was the middle of the session, so I had to come back. And then the first thing we did, that those days people supplied instruments, but they didn't install the instrument. So there was a monster Warburg instrument, and I was there with him, and we set it up, and discovered for the first time 2 respiratory pathways of the mycobacterium. And then he gave me my thesis. That was, so he showed me that how the cell wall synthesis is interrupted, and how the L forms are made, which look like a poached egg, and the complete cell wall synthesis pathway we learned, and we did study the pathology of the pathophysiology of L forms in the very, very initial days of that. And he also made us to put up the protein synthesis complete, make how ribosomes are made, how messenger RNA, transfer RNA, etc. are put up. So in those days, in 1960s, we put up those experiments. And then it culminated in that time, Lederberg got Nobel Prize. So Nobel Prize. So he showed us that how the mutations and auxotrophs are made. So he put us in the science continuously. And one of the acts which I will never forget, we made a cholera vaccine. And he said that, Ganguly, let us eat this cholera vaccine and let us challenge with 569B. At those times, there were no ethics committee. And so what could I say? My teacher, if you had asked me to jump from the mountain, I would have jumped from the mountain. So we took the vaccine and we challenged ourselves 569B. At that time, we didn't know that 569B has residual toxin, but nothing happened to us. We put up all the assays and there were 6 very important paper in Lancet and other journals back to back. I didn't believe in that result and I didn't pursue it further, and he also died. But afterwards, when in the fag end of my career, this particular vaccine was developed by Jan Holmgren, and this was as a DOMI project that John Clements brought it in Calcutta for trials. I facilitated the trials, and this is the current vaccine, whether it is Eurocop, Juvicol, and others, which is now being used in the terms. So Dr. Agarwal asked me he asked several people, that Dr. Talwar to do microbiology, Dr. Mycology, Dr. Mahajan to do parasitology. He asked me to do immunology. I told him that, sir, who will teach me the immunology? So he said, both of us will learn. So you read and I will read and we will learn. And so that is how I became immunologist. Reading and then performing the experiment. So the first T cell experiment and B cell was the rosette. And those many of you might not have put up a rosette experiment, because these are ancient methods of
Rosette and plaque
estimation.
formation.
Plaque
Formation for the B
formation.
cells.
Mehra is not absolute balak at this given moment, so he might have put up rosettes also. But many of you might not have put up rosettes. So that is how we grew in immunology. So the first flow cytometer was purchased by CCMB. And first was purchased by BARC. It was a Partech instrument, was very limited use. It didn't work that well. The second one was CCMB, and CCMB actually was inaugurated by Rajiv Gandhi ji. But they committed a terrible mistake. They didn't have a cooling system. It used to be a monster, fat star. So I purchased the first functional flow cytometer, and because I didn't commit that mistake, and I allowed it to be used by everyone or anyone, because that is one of the things which we need to learn, that if you are fortunate to have equipment, you don't do science with the equipment. You don't publish paper with the equipment, you do science in the equipment. So I allowed everyone to use it, including Institute of Microbial Technology or all the institute, Punjab University, or everybody in the Chandigarh. And they were happy, they grew, and I also grew in this. So you go on doing, and another thing which I learned, that cells are also cells only. So like any other cell. So I started doing systems biology in the immune cells. So could correlate the function as well as the pathway signaling pathways in these cells to correlate with. So went on doing, when the free radicals netioxide came up, they developed those criteria, added on to the interpretation. So go on doing things as the science develops. And when actually the protein and proteome develop, I had 2 persons. The Dr. Luthra from CCMB, who had worked on the crystalline proteins, and Shekhar Mande's wife, Sharmila Mande, who was a protein structure person in our place. And we did work on the prion, started work on the protein association, dissociation. So ultimately, what I want to say, that immunology is a very broad science with multidiscipline. and you have to go on updating your knowledge and learning your knowledge. So when the monoclonals came in the rheumatic heart disease, we produced the B cell epitope of 68 kilodalton antigen and created a test system. And now the Dinender Singla, who actually did that, is now a professor of and head of the department of the therapeutic medicine in Orlando. So this is how we need to really move into this. And one last thing I want to say, that in the immune cells, the matrix proteins are very, very important, because they that creates a platform for the immune cells to act and regenerate. So one of my students, Rajveer Dahiya, who worked on the collagen types, vitronectin, fibronectin, and others interaction with immune cells, is now director of urology in the NCLSC, the USSC in the California University of California. So San Francisco. So this is how the immunology subject develops, and only I didn't learn very well what Mehra learned. I didn't learn the histocompatibility antigen that well,
I
because I
taught you
didn't want
many
to
times.
compete.
I taught
Yeah,
you so many times, you don't
you
learn.
taught me many time, but I thought it is futile to compete with you. I will not be compete with you in that science. But
Well,
but I made one more thing. I made two very beautiful girls to run away from Mehra's lab, because because anyone who entered, Dr. Kanga knows this, because she stayed on. So anybody who enters Mehra's lab, they remained there. They thought but it is very important that you send your students away as soon as they have done their PhD. If you don't send them, their career is destroyed. So I take a great pride in sending those two girls away from his lab by constantly brainwashing them that leave.
Sir,
You
Thank you.
only managed to send two. I sent hundreds actually that you do not know. Anyway, this is really nice. And that was, I was thinking that although I see some questions in the chat box, somebody asking, did you do HLA typing on the responders and non-responders? Somebody asking the negative points about immunotherapy because some patients develop pneumonitis and things of that sort. I think these questions people can really ask him through the email as well. Yeah, I think it is more important to know the history, his own life, and what the youngsters can learn from a career like this, because it is a golden jubilee lecture. And more important than the science that he has showed, of course, quality science that he has showed, is the encouragement of the youngsters. And we are very keen. And I'm a little disappointed with the numbers. We just touched 50 or so. Our society is very large, and we will, they request everybody to participate. And, the onus is on Balaji, kuch kariye, so that the numbers have to increase. Everybody has to increase. You can find a way of giving samosa and chai online if you can. I mean, do something to make sure that the numbers increase. It's very, very, very important there. But I think Dr. Ganguly has had a very long career. You know that the Immunology Society started from AIMS in 1969-70. I had joined as a student at that time, and we created at that time, the first thing was an immunology club of people who were working in immunology in AIMS. So that club ran for about a year or so. And then by early 1972, the society was born. And we had even at that time very few members actually, just about 20, 30 around the whole country. And today we have grown more than 1,000, and I think growing even more. It is very important, as you can see, that the science of immunology has impact on almost all branches of medicine actually, and in biology. And there are a lot of very good biology institutions in India, and there are very good scientists who are working over there. And to people like us who are the beginners during those years, it really gives us a lot of satisfaction to see that the immunologists are now spread all over in various medical and biology institutions in the country. Please raise your hand and ask a question if you want to.
I want to finish, Dr. Mehra, by acknowledging two more persons in my life. I
Yeah.
have acknowledged you, who couldn't teach me HLA very well. So, but I acknowledge
अब
GP.
मैं उसका
I
कारण मैं नहीं
acknowledge.
बताऊंगा, कारण मैं नहीं बताऊंगा, वो शाम का टाइम बताएंगे. तरुणा, you want to ask something?
Yeah, actually, let me acknowledge two persons. I acknowledge GP Talwar, who gave me my first entry as a RAPSAC member when I was very young in the National Institute of Immunology. So that actually changed my life a lot. And I can see Satish Gupta. I am influenced by the work which he has done and the way he progressed and translated things in NII. In NII, I had a huge respect for him, and so I acknowledge him today also.
Can I ask you
Taruna wants to ask, and then Uma, yeah.
Okay.
Thank you, sir. Very inspiring lecture, amazing data, as well as your anecdotes. We have had a full treat. I just wanted to ask a brief question that it is very important, as you said, to identify those 15 to 30% of the responders so that we can be economical and we can save these people from having adverse impacts or no responsiveness. My question is, do you think that there is any chronic fatigue of the immune response in these people who do not respond? Did you see any of the markers which are responsible for the chronic activated fatigued immune cells?
Yeah, actually, the main problem which happens in the tumor is the energy. Massive amount of antigens are released. And another thing which Indranil Mitra actually observed and found out, that the circulating histone and chromatin goes into the bystander cells and causes and all the checkpoint inhibitors or any immunotherapy actually causes harm also, as somebody asked and Dr. Mehra said, that they cause harm also because there are a lot of side reactions which crop up. Not everyone tolerates them very well. So, that is why the medical oncologist is a very important part of such studies, because medical oncologists look at the markers of the side reactions and modulate the doses of the drugs and the spacing of the drugs so that ultimately works. But as you know, some people have to withdraw from the therapy because they can't. And one of my very good friend, Dr. Mehra knows him also, Dr. Gursaran Singh Chatwal, who suffered from cancer and was on immunotherapy, suffered a huge lung fibrosis and died because of the failing lung functions. So I made it look like very nice study and others. It was not like that. And it was so difficult to work with these last 3 years to get 50 patients for 5-year follow-up was a big effort. So the main challenge here was doing analysis and integrating so many parameters together. And here I must confess, I had no expertise in analyzing this complex relationship with the new the programs which have come up in the analysis area. And these are frightfully expensive. I didn't have access to many of them. So many of the friends did it for me. But I took a point to learn. I never allow something that somebody will do it for me and I will not learn. So that was the real crux. So here with these things, we could see that this patient is responding or not responding.
Right.
This could be seen. And that is one of the best thing in this. I think the next generation, I have taken one more student now to look at some of the things which were challenged and I couldn't find answer, and maybe we will find answer to that. So
Sure.
this was just the
First.
first experiment which we did, first study which we did, which showed us where are the frustrations and where we need to get new knowledge.
Sure.
And this new platform in the Karolinska which has come up, I think when we did do the proteomic and this one, because at the moment either you could do the genomic or you could do the transcriptomic or you could do the proteome. You cannot do them together. So this new platform, we can do it together. And recently I was in Munich attending a meeting of this new platform, and about 25 different scientists from different countries presented their data. This is something mind-boggling. Ultimately, but two things come up very significantly in this. Everyone use immunomes because just doing genes and transcriptome and proteome together do not give answer, and everybody did microbiome. This is something which is burgeoning, and they actually modulate the immune response, they modulate the metabolic pathways, they modulate the protein signatures. Like in the nutrition, the Tahmid Ahmad of the ICD-DRB and Geoffrey Gordon did the microbiome and the proteome together, and their paper was published in the journal Science. So these are some of the new science which we have to work, and if we have not learned that, we have to collaborate with someone, because it is not possible to learn everything together. So you have to collaborate with some. So I collaborated with Bhavtosh Das for this microbiome part.
Thank you.
Thank you.
I think we have time only for two more questions. Please raise your hand if anybody wants to. Otherwise, Dr. Ganguly would be anyway available. He's always very prompt in writing back your, well, question. Actually, if you write to him by email. Yes, Uma wants to ask something.
Good morning, sir. Actually, my question had been answered when Taruna asked the question in between. But I want to salute, sir, for his contribution and want to ask you, sir, one thing, that what was your most exciting finding that excited you a lot during your research career? So,
Every finding.
Uh, my main passion was in the rheumatic fever, rheumatic heart disease, on which I worked whole my life. And that was the passion, but we didn't find a solution to that, unfortunately. But majority of my work has been devoted to that. It's an autoimmune disease, so this was one of the immunologic profile which was very exciting. So I have been able to walk with this science with as it was, and Dr. Mehra was also associated at one time with this group, and ultimately the lot of recognition came, and this one so this is one which excited me in my life. And if you go on doing, although when you receive a lifetime achievement award, people tell you that you are no good anymore, so take this award and disappear. So this time I got when I was in United States, so in the two major associations with which societies with which I was attached, they gave me as a surprise lifetime achievement award. So this was very significant for me. But I hope nobody gives lifetime achievement award to people, because that shows a signal that disappear.
Yeah, he talked about
Or
rheumatic fever, rheumatic heart disease. I do remember in 1983, we published a paper on D8 by 17 and 883 independently of the Chandigarh group, and our findings were nearly similar. in terms of the presence of this alloantibody on the patients with rheumatic fever, rheumatic heart disease. There was this guy, John Jabrisky, in New York Medical Center. He had discovered this alloantibody on the patients, and our findings were very, very similar there. So I also agree with you that that disease was very, very exciting. Somewhere down the line, and we of course continued our work on rheumatic fever, rheumatic heart disease, publishing papers on HLA association, things of that sort. But somehow, I think after the departure of Dr. Wahi and people like that, and Dr. Bhatia here, the interest in this field had gone down.
Many people, Dr. Mehra, Rajan Tandon,
Rajan
फिर
Tandon,
Dr.
of course, and
Srivastava, all of them
names.
continued in all
And
India
आपके
through
यहाँ
leadership.
वो
But
जो
but the
सरदार जी,
but the numbers decreased so much,
yeah,
and this is one of the neglected disease over here in India. So we still haven't found solution. So in future, I hope somebody takes it up. Now Dr. Karthikeyan in the All India Institute follows this. There are some people who are following it.
yeah.
सर और हम डिपार्टमेंट में रह गए, हमने भी बहुत कुछ सीख लिया डॉक्टर मेहरा से. आपने कुछ लोगों को भगा दिया, बट आई लर्नड अ लॉट फ्रॉम सर. सो
आपको नहीं भगा पाया.
नहीं, बहुतों को नहीं भगा पाया, ऐसा नहीं. अब बहुत भागे भी, ऐसा नहीं. Anybody, last question, because we don't have any more time. Who wants to ask? And otherwise, we thank Dr. Ganguly from the core of our heart. Thank you very much.
Thank you.
And we also thank the Indian Immunology Society for starting this very important series of Pratiraksha Swaran Jayanti Vikyan. And the third one will be held next month. So they're going to do every month, which is a very, very good thing. Thank you very much, Dr. Ganguly, for
thank you,
taking
Dr.
time
Mehra.
out from your
Thank
busy
you,
schedule.
Taruna. Thank you, everyone. Thank you.
Thank you. Thank you.
Dr. Bala, may I invite you to please say a formal vote of thanks.
Good morning, everyone. First and foremost, on behalf of the Executive Committee of Indian Immunology Society, with immense gratitude, we express sincere and wholehearted thanks to our eminent speaker, Padma Bhushan, Professor M.K. Ganguly, for delivering the prestigious second Indian Immunology Society Golden Jubilee Lecture and sharing his valuable big data from clinical trials to immunotherapy of cancer patients. Sir, it is really an exciting moment for all of us to listen to you, and also inspiring for the younger generation, since you shared all your experiences. We really honored by your speech, sir. We express our heartiest thanks to the chairperson of this event, Professor N.K. Mehra, former Dean, All India Institute of Medical Sciences and Vice President, INSA, for having orchestrated this august function in a scientific way. Sure, sir, we will take necessary steps in the future to attract more students and researchers to listen to the, experienced speakers like Ganguly, sir. We thank all the participants across the country for their overwhelming response. Thank you all, including all the senior immunologists who spared the time. We place on record our sincere thanks to the IT team, ICMR NIRRH, and for their seamless work in making this event a grand success. Thank you one and all. Thank you all.
Thank you very much. Thank you. Bye-bye. Namaste.
I just want to make an announcement. Sir's lecture would be available all the time on the YouTube link that we have provided. So you can listen to him again and again, anytime when you want to. Thank you, sir.